Okay, I think we will get started. Welcome everybody to day 2. My name is Holden Maecker. I'm director of the Human Immune Monitoring Center, and I'm going to give you a little introductory lecture this morning about the HIMC, and then we will get into some additional exciting lectures about phosphoflow and CyTOF. So welcome, and feel free to ask questions as we go. This one will be pretty short, but I wanted to sort of give you an introduction to what we do in the HIMC. It's kind of a unique core facility that we've built at Stanford. Basically, this was the kind of brainchild of Mark Davis a few years back, who wanted to create a service center that was like a one-stop shop for immune monitoring technologies. And so we've tried to basically help people with everything from the design of their clinical study and of the assays that they should choose, to sample processing and banking, to actually running the immune monitoring assays on multiple different platforms, which I'll talk about, and even data analysis and helping them with the interpretation, biostatistics, etc. So that's kind of the major goal of the center, is to provide this sort of full-service immune monitoring help. But then we also have a sort of secondary goal, which is to be a sort of testing ground for new technologies and to adopt new platforms that might be useful for immune monitoring. And a third sort of goal is to actually amass the data that we're generating. As long as we're using the same standardized assays in multiple studies, we have the ability to actually compare those and bring them into larger data sets. And so we're building some tools with which to do that, and I'll mention those briefly. So one of the driving forces that we try to abide by is to do comprehensive immune profiling. And so if you're going to do this, you're going to offer all of these different kinds of immune monitoring assays, you've got to do some picking and choosing, right? And if you're going to be standardized, you can't standardize every assay under the planet. And so we've tried to standardize the assays that are sort of most comprehensive, that will allow the largest number of people to come and get something that they want from it. And the comprehensive nature is sort of in a couple of respects. One is that we're looking at multiple cell subsets, proteins, and genes. So we're at different levels in the biological hierarchy, if you will. And so we do microarrays that are now very common, but that span the entire human genome. We do a Luminex assay for cytokines and related proteins, which has now 51 analytes in it. And we do flow cytometry phenotyping for 44 different subsets, but we're now moving to CyTOF, where we can actually have a huge number of subsets using all of the same markers in a single tube. Yeah, so Luminex, I'll show you, is sort of a bead-based immunoassay. So very much like an ELISA, but it's multiplexed with many analytes in a single well. And then a second sort of driving principle that we have is to try to do assays that look at stimulated responses. And so in addition to just measuring the resting phenotype, as I'm sure you heard yesterday, you can uncover a lot of defects that you wouldn't otherwise see by doing some type of in vitro stimulation. And so in that regard, we do phosphoflow, we do intracellular cytokine staining, and we can also do both Luminex and gene expression arrays from stimulated cell samples as opposed to just from serum. Now, one of the consequences of setting up this center and offering this kind of full-service immune monitoring is that we've actually gotten the interest of a lot of different kinds of groups on campus and some outside of Stanford as well. And I think it's sort of an interesting commentary that what we're doing really is making immunology accessible to non-immunologists. And so this is a kind of a smattering of projects and and different departments and divisions that we've worked with in the last year or two. And you can see that it's things you wouldn't necessarily think of as immunology-based projects. So it's not necessarily autoimmune disease or immunodeficiency or something that's really pure immunology. We have those projects too, but you know, there's a group that's looking at the effects of opioids on cell signaling in wound healing and in settings that are of interest to anesthesiology. There's somebody looking at sleep deprivation, and believe it or not, that affects cytokine levels. Maybe not too surprising, but I think it's great that we are starting to enable immunological studies in some of these areas. So quite a diversity of places where immunology and inflammation play a role. We're also starting to do some clinical trials, so actual outcome-based studies in which there's an intervention and people are looking at either a new drug or some type of immunotherapy, for example. Some of them externally sponsored, some within Stanford. They're generally phase 1, but there's 1 or 2 phase 2 trials that we've worked with as well. So, you know, we're kind of bridging that area. We're not really just out there to do clinical trial monitoring. We want to do much more than that, but we do have a number of clinical trials underway as well. Now I want to just briefly tell you about some of the technologies that we use as standard assays in the HIMC. You've of course already heard a lot about mass cytometry, and we are adopting that now, as I said, to do our immunophenotyping. We're also doing intracellular cytokines by CyTOF, and we're starting to do phosphoflow as well. We have, however, a standard phosphoflow assay that we use by fluorescence, in which we use a battery of 8 different cytokines, and we read out multiple different cell types and multiple phosphostats in response to those cytokines. So that's a very nice standard assay that we use to sort of probe the cytokine signaling networks. And then we have some assays that are at the protein level. The Luminex, as I mentioned, in which we can analyze 51 different cytokines from serum or plasma. And maybe I should have put a little bit of an explanatory kind of figure in here, but basically this is a bead-based assay which captures a different cytokine, in this case, on each different bead. And we have a series of 51 different beads that are separated by a flow cytometry type of instrument. What this does show is the sort of reproducibility of the system using a control serum sample that we include in every run. And this is just an example of 5 runs over a period of a couple of weeks where you see the raw data for here 25 and here another 25 cytokines, or 26. And it's pretty consistent. And then I looked at this last point and I said, you know, it looks like this last run is not quite in line with the others. There's more cytokines that have differed. And I went back to our immunoassay manager and she said, oh, that's where we switched batches of the control serum. So at least the assay is sensitive to that. Now, we also have a kind of a unique platform for doing a kind of nanoscale immunoassay, which is called NanoPro. It's from a company called Protein Simple, and you will hear more about this tomorrow in both the lectures and demos. This is a capillary isoelectric focusing platform which is automated and which allows you to identify proteins of different isoforms. It could be phosphoisomers, or it could be other kinds of modifications that change the PI of the protein. And so you can detect those as peaks, and you can look at phosphorylation, for example, in that way. And it's very useful for small volume samples. You can lyse down to, believe it or not, a single embryo and get a signal on this instrument. So we've actually done a project where we looked at single mouse embryos. With cells that don't have that much protein in their cytoplasm, you may need a lot more. So PBMCs is more like maybe 10,000 PBMCs, but you can get signals from small numbers. And then another platform that you will hear about tomorrow is the Fluidigm platform for doing qPCR. And so these are our mainstays of gene expression assays, qPCR and microarray. We do a variety of different kinds of microarrays in 1 or 2 colors. And we actually do some mouse work, but not a lot, because we are the Human Immune Monitoring Center. And then we have a battery of data analysis techniques that we are implementing and expanding. And one of these that you'll also hear more about tomorrow is this cell subset specific deconvolution of gene expression data. And so basically it's a way to take gene expression data at a group level, which you've derived from whole blood, for example, or some other kind of mixed tissue, and for which you also know the frequencies of the various cell types in that mixture. So if you've done some kind of phenotyping of the whole blood at the same time point where you've done the gene expression, you can then mathematically deconvolve the relative expression of each gene in each particular cell subset. And Rob Tibshirani will tell you a lot more tomorrow about how that works, but it basically allows you to make comparisons at the cell subset level, even though you did your analysis on whole blood. You've also already heard about SPADE, and we're beginning to implement this in our CyTOF assays. So we have a sort of standard way of looking at the cell subsets that we differentiate in PBMCs by CyTOF. And I'm always impressed that these sort of SPADE trees that are generated sort of recapitulate what we generally know about differentiation of T cells and B cells to quite a consistent degree. And so here is expression of CCR7, which is really restricted to one branch of the CD4 T cells. So I'm calling it the central memory branch. It's much less expressed on this other branch, which I call effector memory. And similar with CD8 cells, although there's a gradient along this branch, basically the high expressors for CCR7 are all out on this one limb of the tree, and there's another branch of CD8 T cells that is basically not expressing CCR7. So it's very reproducible and sort of expected distribution of these markers along these branches. And then we have NK cells, monocytes, and B cells over here. Now, I mentioned that we're also trying to amass this data with tools by which we can explore the data even across projects. And to do that, we've been working with a contractor called Cytoanalytics, and we're building a system. We've already built a pretty good version of it called Stanford Data Miner, which is basically an online tool to allow data exploration and data aggregation. And it works by basically allowing you to construct tables online and choosing the dimensions that you want to display in those tables, and then also choosing filters to apply to them. So we call these displays cubes because they're two-dimensional tables, and the third dimension is the filters that are applied to the table. And so here in this very basic cube, which is not expanded in any dimension, you can see that there are 3 different kinds of assays displayed in the rows, and gender is on the columns, although it hasn't been expanded. And there's a couple of different measures being shown here, the mean and the number of assay points for each of these. And so all you can see from this cube is that flow phenotyping, Luminex, and PhosphoFlow were all done on these samples, and this is how many data points there are for each. If you clicked here to expand the genders, which is what has happened here in the bottom display, you would begin to break these out, and it would calculate for you these statistics on the fly so that you could see the rolled up means. And in this case, we've changed the display here to also show min and max for each of these categories. And now what we've done in this lower panel is also expanded the analytes for Luminex MSD and chosen only 4 of them just for simplicity So now you can actually see data that is more meaningful, and you can see that males versus females, there might be a little bit of a difference in the level of CD40 ligand. Possibly for interferon, it looks like interferon gamma is higher in the males than the females in this particular data set. And what you can further do then is you could click on one of these numbers and see all of the individual underlying data so that makes up that mean. And so you could start to ask whether there are outliers that contribute to this high number here or this lower number here, and start to explore the data in that way. There's also some rather simple graphing functions that come along with this so that you can look at it graphically as opposed to numerically. But the basic idea is to be able to explore your data and bring heterogeneous types of data together with what we would call metadata, the clinical and demographic information that comes along with your study. So that's basically what I wanted to tell you about. These are the people that do the work in the HIMC. We worked in collaboration with BD for some of our CyTOF development and cytoanalytics for the database that I showed you. And we are part of this Institute for Immunity Transplantation and Infection, which is the sponsor actually of this meeting, and which is run by Mark Davis. And so we enjoy a lot of collaboration with Mark and with Gary's lab to make all these kinds of assays possible. So that's basically the extent of it, and I'm happy to take some questions if anybody has. Otherwise, we're a little ahead of schedule and we can move into the other talks. Questions? No? All right.